Hey everyone. My name is Jessica Ma, and thank you very much for tuning into my presentation. I understand that I am the last session of the day, so an extra thanks for sticking around and waiting for this talk. What I'm going to do today is talk about some of the work that has recently been done in our lab. The title of my presentation is Identifying Transcriptional Regulators of Human Embryonic Development via Expression Variability. And I'd just like to remind you guys, if you have any questions along the way, please feel free to submit your questions, and I'd love to answer them at the end of the presentation. So let's go ahead and get started. The focus of this talk is around transcriptional regulation of embryonic development. Now, we all know that as an embryo, there are various key developmental stages that these embryos transition through to develop normally. And a big part of regulating that transition is how the transcriptomes are controlled. So there's a paper that came out at the end of last year in Nature Structural and Molecular Biology from Yan et al. And they were the first to produce a data set based on single-cell RNA-seq profiling of human embryos and embryonic stem cells. And so this was a really exciting advance, which allowed us to really look at how transcriptomes were changing during embryonic development. Now, just to step back a little bit, the focus of my lab here at Albert Einstein is to really focus on how genes are regulated through transcriptional regulatory networks. And as we know, genes are expressed with both signal, but as we're starting to understand as well, the level of precision or the degree of noise which these genes are expressed also has functional implications. So if you think about what's going on within a single cell, and you think about how components of a regulatory network are controlled, you can imagine that genes with low variance or low variability in their expression are more uniformly expressed between cells. So if we were to sample any cell at random from a population, the chances are that level of a particular gene is highly predictable because it's quite similar from cell to cell. At the other extreme, you can imagine that genes that are more variable or have much higher variance in their expression reflect greater heterogeneity, so that if you're looking at the population of cells, these cells adopt different levels of gene expression for this particular gene. And you can imagine then the functional consequences downstream depending on the roles that these genes are involved in. So another way to think about gene expression variance is as a population-specific parameter. And this is something that our lab has been doing for a little while now, where you can think about two groups, usually a disease group and a control group, that you're interested in contrasting phenotypically. And what we tend to do is we look at gene expression differences from the perspective of averages. And we identify genes that are differentially expressed based on how different they are in terms of average expression in the disease group and the control group. And in this setting, where traditionally we use a t-statistic, in this case, the variances between the two groups will be the same and likely minimized. So this is a very sensible way to identify elements that characterize a phenotype. But one aspect that we're missing out on is the ability to identify genes that have different degrees of variability between these two groups. So one example would be, for instance, that's shown at the bottom of the slide, is where the expression of a gene is quite widely distributed in the disease group, and in contrast, quite precisely distributed in the control group. And a more extreme example of this is where, on average, the expression of this gene is the same in the disease and the control group. But as you can see, the variability or the variance of this expression is quite different. Now, using traditional approaches like the t-statistic, we would miss out on identifying these genes. And so a lot of our work has gone into trying to make sense of how variability and variance can help us understand regulation of disease and different phenotypes. So another kind of motivation, particularly in looking at single cells, is this realization that when we drill things down to the single cell level, gene expression is actually reflects as discrete states in the population. And a way to demonstrate this is work that we've done with our collaborators, where we've taken, or they've taken rather, single cell gene expression from the HES3 cell line. And what they've done is they've sorted these HES3 cells according to a cell surface marker, CD9, which represents the degree of pluripotency. So what we have are these FACS-sorted different subpopulations of the same cell line that reflect different degrees of pluripotency. And you can see as you move from left to right on the slide, we go from increased to decreased levels of CD9 protein expression. Now, because we have the level of protein expression for this gene, we can also look at the actual gene expression or transcript distributions of the CD9 gene. And when we look at the individual single cell profiling, which we've obtained using Fluidigm Biomark, what you see for the 100 cells are these discrete distributions. So for instance, and I should point out that this is CT data, so as you probably know, the higher the level of CT, the lower the degree of expression, and the lower the CT value corresponds to genes that are more highly expressed. So what's really interesting here is you see the discreteness of CD9 transcript expression as you go from high to low levels of protein expression. You can see that for the highest degree of CD9 protein expression, we see 100% of the cells in this high transcript expression state, and contrast that with the least or the lowest level of CD9 protein expression, where you see that the majority of cells are actually in this off state where there's no transcript expression of CD9, and there's a small subset of cells that are expressing CD9. And everything in between is kind of interesting because it's this balance between genes that are in the off state at a CT value of 40 and another subset of cells that are actually expressing the CD9 transcript. And as you go from high to medium to low levels of CD9 protein expression, what we're seeing is this transfer of cells moving from the on to the off state, as opposed to something like a bell curve that's shifting on average to lower degrees of CD9 expression. So this is a real plug, I guess, for looking at gene expression distributions and by proxy variability in single cell gene expression, because if we just looked at average expression, we'd miss these subgroups that are clearly there in the population. So let's go back to human embryonic development and talk about the data set that we've been using from Yan et al. What we've got here from the paper is, as I said, a single cell RNA sequencing data set. There's about 20 to 60 million 100 base pair reads per cell. There are 3 embryos for each developmental stage, except for Morulae, where we had 2 embryos from the paper. As far as they state, all embryos were morphologically normal on appearance. And so basically what we're working with is an increasing number of cells for 2 to 3 embryos at each developmental stage. So if you have a look at the data from an overall perspective, we're working with the RPKM normalized data. There were some filters that were passed to ensure quality of the data, and we've adopted them in our analyses too. We've taken a threshold of 0.1 to retain the genes that were expressed or more highly expressed, and we've required that that be the case in at least 75% of cells from the same stage. This gives us about 8,000 genes to work with in our analysis, and this is spread across about 120 cells. So in terms of measuring variability, there are different ways we can do this. And in our analysis, what we've done is adopted 2 different statistics to understand how variability is occurring in expression at the level of cells and embryos. So if you imagine that X is the expression of a gene in a specific cell in a specific embryo, what we've done is we've calculated the SDC, which represents the intercellular variability in the one embryo. So we can imagine that STC represents how much the gene expression fluctuates for the same gene from cell to cell within an embryo. And basically what we do is we look at the intercellular variability by averaging this STC that we've obtained from one embryo over the multiple embryos that we have available, so that this is the best estimate that we have for intercellular variability. We can also look at heterogeneity or variability within the embryo as well, and for that we calculate the inter-embryo variability, where we're looking at how this STCJ statistic fluctuates about the mean STC that we can obtain for the 2 or 3 embryos that we're looking at. In the course of our analysis, as you'll see, we're actually a lot more interested in the STC, but if we wanted to, we could also study the STE. So if we step back and think about these statistics that we've described, we ask obviously the question of what can this variability tell us about a group or population of cells. Well, if you think about STC, low levels of intercellular variability reflect stable invariant expression. Okay, the gene is expressed homogeneously pretty much amongst the cells that we're looking at, and as a result the level of expression is highly predictable, so that if you were to sample any cell at random from the embryo, then we have a lot of confidence in predicting what that particular gene expression level would be. If we contrast that and think about genes that have high STC or high intercellular variability, then this reflects genes that have widely distributed or variable expression. And in this case, we think of a population of cells, and this gene being expressed heterogeneously from cell to cell. And as a result, then if we sample at random from this population, we have less confidence in predicting what that gene expression level should be. As we saw before, a diversity of different expression states could actually lead to cells that are undergoing transitions to different cell fates, or maybe different phenotypically for other reasons. So if we look at the global, and by global we mean across all genes that we're looking at, we can see that intercellular variability actually increases across development. So you can see the STC, the distribution is basically increased as we go from 4-cell to blastocyst stages. Now this is obviously probably not much of a surprise to anyone. We know that as embryos develop, the cells become much more heterogeneous because they are undergoing specialization to different lineages and tissues and cell types, etc. So it's interesting to see that reflected in the overall distributions of the genes. If we look at STE, as you recall, measures how consistent the intercellular variability is between the different embryos. And we can see that the STE is largely constant, or the distributions are fairly overlapping as we go from 4-cell to blastocyst. So in a way, this gives us confidence. It means that the embryos are fairly stable, if you like, and it means it's a good reason to study the intercellular variability. So if we look a little bit more carefully at the distributions as the embryos develop, we notice too that the global distribution of intercellular variability spreads out. And here in these densities, what I've shown is the density of the STC and the STE, where each point on the density represents a gene. And the round dots that you see at the bottom of the graph represent the average expression variability for that particular developmental stage. So the STE, you can see all the average expression variabilities are actually sitting on top of each other, which is reflective of the lack of difference or the lack of shift that we see as the embryos develop. In contrast, the STCs, you can see that even on average, there is a consistent increase that we're seeing in the distributions. And what's interesting too is as the embryos develop further, we see a widening of this distribution, which means that there are some parts of the genome that have very stable expression, and there are other parts of the genome that have high variability from cell to cell in the embryo. So, Oh, there's my animation. Okay, so our interest, therefore, is to look at these contrasting regions of the genome and ask what genes are these and whether specifically they target different pathways or processes that require this difference in variability. So to do this, we applied a mixture modeling technique using normal mixture models using the mCluster package. And if you're not familiar with mixture modeling, basically what this is decomposing a specific density into subcomponents that we can then explain. So for instance, on the left, I've shown this cartoon where we've identified 3 components, and each of these components then corresponds to a subgroup of genes in our particular setting. And what we've done then is to apply this to each of the densities we obtain for each developmental stage. And you can see that for the SDC, for the 4-cell stage, we have 4 components. The 8-cell stage, we have 3. For the Morulae, we have 3 components. And for the blastocyst, we have 2. So what we want to do then is to identify whether these subgroups correspond to an enrichment of certain pathways or processes. And to do that, we're using mTOR and Ingenuity pathway analysis. These are 2 software tools that have annotation terms and functional annotation terms to identify different kinds of pathways involved. So what we do is we identify first the subgroups using mixture modeling, and we look at the overlap and assess the significance of overlap with specific pathways from IPA and mTOR. And we assess the significance using Fisher's exact test and adjust multiple testing using false discovery rate corrections. So the results that we see are actually quite interesting. For the 4-cell stage, we see that there is a distinction between pathways that were enriched at the most stable group versus the most variable subgroup. And for the most stable genes that had the most stable variability, the most stable expression, the lowest variability, we see pathways related to critical processes like cell cycle, translation, and transcription. On the other end, when we look at the genes that had the most variable expression, we see pathways that are related more to metabolism. So for instance, in OXPHOS, we see we also see pathways that are related to apoptosis and cell survival. So this is really interesting, and I just wanted to point out too that all terms that we've reported here are significant at a 0.05 p-value, and they're coming from mTOR, although we see similar results in the IPA analysis as well. This kind of cell cycle versus apoptosis metabolism result is something that we see persisting through the different developmental stages. So for instance, at the 8-cell stage, for the most stably expressed genes, we also see pathways related to cell cycle, and here we see some related to DNA damage as well. For the most variable subgroup, we see one apoptotic pathway that's significant for this group of genes. For the Morulae, for the least variable subgroup, again, we see these cell cycle terms, and for the most variable group, we actually do not see any significance in the terms, and that just means that the genes that were in this component that were identified were in just so many different kinds of pathways and functions that they weren't significant enough to point to a specific trend or enrichment. And finally, for the blastocyst stage, we have 2 components, and if you look at the most stable and the most variable, it's almost like we have a flip of the previous pathways. So for the most stably expressed genes, we see an enrichment of, again, metabolic energy generating related pathways like OXPHOS, glycolysis, and gluconeogenesis. For the most variable genes, we actually see the cell cycle and the DNA damage pathways come up. And we're not sure really what that is reflective of. Perhaps these genes, or these cells, are now specializing into other roles, and hence the reversal of these energy versus sort of cell survival kind of pathways. The other thing that we've done in this analysis is to identify new markers of each developmental stage using measures of variability. And if we think about how we tend to find markers based on expression, this tends to be based on average statistics. So we look for, on average, an increase or a decrease in expression between the phenotypes that we're contrasting. But when we think about what's going on in single cells, as we saw before, looking at averages may not be the whole picture, and we may in fact be missing important shifts in expression. So I've just drawn a cartoon here that reflects what's going on, in the sense that in an intermediate stage where we're trying to identify markers for, it could be that we see a massive shift in the precision of expression of that marker, but on either side of that stage we see a really noisy or heterogeneous degree of expression. So we asked the data whether we could see genes that fit this profile, and in doing so what we required was genes that had a significant change in variability across all stages. We used the Levene's test to test this. We also required that the STC for that particular stage be less than the STC at all other stages. And we also looked for an increase in fold change for that particular stage compared to all other stages. And as you can see from the box plots, we do find genes that fit this criteria. So if we look more carefully at the line profiles, which is probably a better way to view this, we found 55 potential marker genes for 8-cell stage, 8 for Morulae, and 11 for blastocyst. And just because we're running out of time a bit, I just wanted to highlight really briefly what these profiles look like for these potential markers. Now you can see that for the Morulae stage, these genes are expressed quite tightly, and I'd just like to point out each dot in these plots represents a different cell. And one of the really interesting genes here is MESDC2, which is a mesenchymal chaperone protein for stem cell patterning and development. And so this has an obvious role in regulation of the embryo. And what's interesting is some of these other genes also have interesting roles, some of them obvious, some of them not. But some genes are also lacking information on what they do in the genome. So it's interesting to pull out more information and sort of infer how they could be potential markers or potentially important to embryonic development. When we look at the blastocysts, here we found 11 genes. And again, some of these are functionally very interesting. One of them is EpCAM. EpCAM you've probably heard a lot if you work with stem cells, particularly embryonic stem cells. EpCAM is a potential marker for blastocysts, and it's involved in formation of the trophectoderm. We also find some other interesting genes, and we're still in the process of exploring their contribution to embryonic development and their utility as a potential marker of stage. So we've done some analyses focusing on how changes in variability are important, but another way of looking at this is also to ask if genes that have invariant expression or very stable expression, are they also important to helping understand how expression is regulating embryonic development. And here I have to apologize for the typo in my slides. X is about 6,000, so we found about 6,000 genes that did not have significant change in variability. And again, for this we used the Levene's test and took those genes that were not significant at the 0.05 level. So what happened was we took these 6,000 or so genes and we clustered the variability profiles across the different developmental stages to identify what kinds of patterns were present. And as you can see from the cluster profiles, basically these fall into 3 groups that reflect low, medium, and high levels of stable expression. And so what we did was to then focus on the genes that were in the most stable group. So these guys had the least variability going from stage to stage, and also had the lowest level of variability compared to all the other groups. So when we look at their corresponding expression levels, we noticed something really interesting. We saw that basically these genes fell into three modes of expression, which we've termed off, on, and super on. And so you can see at each stage this trimodal distribution is apparent, and you can see how off corresponds to zero, or very low average expression. On corresponds to like middle level, but certainly moderate to high levels of expression, and super on corresponds to extremely high levels of average expression in the embryo. So to define these three groups of off, on, and super on, we use normal mixture models, as we did before, to identify which genes were being classified into one of those three groups. And then what we did was we looked at mTOR and IPA to look for enrichment of specific pathways and processes at each developmental stage. So when we look at the on genes, we actually found that there were quite a few genes that were commonly classified as on in all four stages. And so when we look at the pathways and processes that were enriched in this group, we notice that there are some really critical pathways that are involved in signaling and survival. So for instance, EIF2 signaling, mTOR signaling, ubiquitination, cell cycle, and nucleotide excision repair are all represented amongst these genes. When we look at the on genes at each stage, we actually see there's quite a lot of overlap in the terms that were significant. And we can also actually see that there were some terms that were specific, and these are indicated by the stars that you see. When we look at the off genes at each stage, we also notice overlap, and the trend that's emerging here is that these are terms that deal largely with differentiation and development of specialized cell types. So it makes a lot of sense as to why embryos would prefer to turn these off in the cells and consistently amongst all cells, particularly as the embryos are at a very early stage of development. And then the super on genes that we see are, there are fewer enriched terms here, but the terms that we do see are quite interesting. And we see for the first three stages this consistent term of differentiation of embryonic stem cells. And this makes a lot of sense, I guess, given the type of cells that we're looking at. For the blastocyst, we also saw a term related to NADH repair. And so this suggests perhaps that there is an energy demand which we know is increased in blastocysts. And so perhaps by turning these genes on consistently at high levels in all cells, this might be supplying an increased energy demand that's coming from the blastocysts at this stage of development. So I'm going to end my talk there by just summarizing some of the bullet points of our analysis. Using variability and also average expression, we've been able to identify some putative markers for each of the developmental stages. And some of those markers seem quite plausible in terms of what we already know about how they contribute to regulation of embryonic development. We've shown that globally, variability increases as the embryos develop, which is in line with our understanding of differentiation and cell type specialization. We've also highlighted how distinct pathways and processes are under different levels of regulatory control as these embryos transition through development. And we've been able to identify too the pathways and genes that are more stably expressed, and how these make sense hopefully to understanding how embryos develop. So I'd like to end there and acknowledge the people who are involved in this work. Yu Hasegawa is a student in my lab, and she's done a majority of the analyses that I've described. We've also been working with other people in my group who have contributed in various ways. We collaborate with Dr. Deanne Taylor and her postdoc Jessica Landis, who are at Reproductive Medical Associates of New Jersey, and they've been really instrumental in guiding the analyses that we've described. So I'd like to end there and thank you all for your attention, and I'm going to just click on this Q&A box here and answer the questions that have been submitted. So the first question comes from Maria, and she says, you mentioned about enriched pathways and functions. Is there any reference related with this point? If you're referring to the method, Maria, there is a lot of literature, I think, associated with this kind of enrichment analyses. I'm just blanking at the moment with I guess you could look at different kinds of software packages. For instance, GSEA is one related program. It's available from the Broad Institute. There's also a package called David, which is not what we used, but this also is a freeware tool. I'd encourage you to have a look at it. It's actually really, really helpful, and there's references associated with that. There are references associated, I think, with mTOR and IPA as a tool, and I think a quick Google PubMed search would also lead you to what those are specifically. If you mean references related to the pathways, the enriched pathways that we discovered from this analysis, these are there in the analysis that we got back. And we're in the process of looking at some of those references, in particular the PubMed IDs as well, that reference the work that gave rise to those annotations from the literature. The next question, also from Maria, is, was there any activation with X chromosomes? I really don't, I don't know if we have the data to analyze that. I guess we could look at transcription from genes on the XY chromosomes, but we only have RNA-seq, so I'm not sure about how to answer that question. The next question we have is this a one-time point expression profiling, or over a period of time? Is expression consistent for particular differentiation or developmental stage? That's a great question. The expression profiling was done over a period of time. However, it's important to realize that we aren't following, or we aren't able to follow, the same embryo over each developmental stage. So we are looking at snapshots of distinct time points, but they are supposed to represent a longitudinal kind of change in expression. It just isn't on the same set of cells. And is expression consistent for particular differentiation or developmental stage? We do expect we do expect to see specific, sorry, the question just disappeared. We do expect to see consistent patterns that represent each developmental stage, but obviously there's a lot of variability and heterogeneity going on. So separating out that signal to noise is what we're also trying to do. Okay, so the next question is it possible to have a sample for your future publication about your results? Yes, that's an excellent question. We're in the process of writing this analysis up. This has not been published yet, but we definitely hope to have that publication out soon. And our last question deals with, again, with X chromosome. Women suffered inactivation on one X chromosome, the other chromosome X is active. Is that something, I'm not sure, Maria, I think it sounds like you may know more about this than me, but if that's something that we can see transcriptionally, then we could look for like a dosage effect. I do think, though, we would still need to look at more embryos than the number that we have available to be able to study more concretely the contribution of the X chromosome. So I think these are all the questions. So I am going to end there, and please feel free to follow up with questions via email. I'd love to receive them, and thanks very much for your time.